Okay. Good evening, guys. I thought I left the last video was an MLPA where I ended the question with: is it possible for next generation sequencing to diagnose a case of Duchenne's muscular dystrophy? I left it there, and the answer was no. I said why it is no, I'll cover in the next video. So today we will be seeing two important topics: next generation sequencing and genome wide association studies. This is otherwise called as genome wide association studies, GWAS studies. Questions have not been asked from this session, but I strongly believe that in the coming central exams, at least one of the exams will have questions based on this, because the future is going to be this. Fine. So what is next generation sequencing, or NGS, or massively parallel sequencing means? I'll just simplify the concept. Actually, it's an oversimplification of the concept. The real complexity of next generation sequencing is much more than what is there in the video, and there are few intricacies which I'll be skipping, which are not required for an MCQ purpose. First, what is sequencing? Before going for next generation sequencing, we have to know what is sequencing. Sequencing is, okay, for example, we all know cystic fibrosis is a disease mutated in gene CFTR. So this has a sequence like A, T, C, G, blah blah blah. So this sequence will give rise to mRNA, which will give rise to a protein, amino acid and a protein. We all know the most common mutation in cystic fibrosis also. So this is the sequence. This is the sequence. So which caused the change in amino acid is one base pair most of the time. So from a patient of cystic fibrosis, if I am able to see all the nucleotides which codes for the gene, that is called as sequencing. That is called as sequencing. Like a cystic fibrosis patient comes to me, I take his blood and I do some method and I say his cystic fibrosis gene is ATCGGGAT, something like this, and this point actually it should be seen, now it is mutated, that's the cause of disease. That is called sequencing. So for me it is easy to sequence single gene. One gene, yeah, it is okay to sequence. Two genes, yeah, it is okay to sequence. But yeah, if I have to sequence all genes, that is entire body of mine, the genome of mine, how do I do it? Single gene sequencing will not be possible. It will be very tedious. It will be very damn expensive. That's why this concept of next generation sequencing came into place. I'm saying again, I'm oversimplifying the concept. The exact thing how it happens is we will have this as the genome. Okay, we'll have this as the entire genome including exons and introns. Okay, so what I do in next generation sequencing is I take the blood. It's done from peripheral blood. This can be a question. It can be done in peripheral blood from any source which can get a DNA. It can be done from abortus also. It can be even done from saliva. It can be done from any source which gives a DNA. I extract the DNA and there are primers involved and amplification involved. If the question, what is the technique behind next generation sequencing, it is PCR based amplification is the primary one, and then we sequence it. Amplification is the primary technique. Fine. So now I have this patient's DNA like this. Okay, this patient's DNA is here. So we make it a single stranded DNA. Now what I do is I cut these DNA into small bits and pieces. Okay, I cut this DNA approximately 200 to 250 base pairs. I cut the DNA into different pieces. This step is very important. I cut the DNA into pieces. So like for example, a 1 meter DNA is being cut into multiple pieces, so it will be something like this. All through the genome it will become like this, into small pieces. Okay, so now what do I do is, the next step is going to be amplification. I amplify each and every segment all throughout, each and every segment all throughout. Fine, now I have amplified it and I've got something like this. I don't know which gene it is, I don't know which chromosome it is, I don't know which cytoband, PQ arm, nothing. I don't know nothing. I know nothing here. Right? But the beauty comes here is what? The computer is fascinating, at least for me. What computer does is, it is in 200 to 250 base pairs, right? Fine. So we know a normal human genome's code, normal, or we'll keep it as a library. We know already a normal genome library, which means what is there from chromosome 1 to chromosome 22, X and Y, what is there, A, T, C, G, entire thing we know, that is called a library. What this beautiful computer does is, it matches it. Yeah, this base pair goes here. Okay, this base pair goes here. So if it's chromosome 1, chromosome 2, okay, this may be chromosome 18, it goes here. It matches these things here and here, right? So finally it'll give me a data. Okay, this is the entire sequenced product. So I'm repeating again. I take a DNA, I split it into single strand, I cut into million pieces, and all these million pieces are amplified parallelly, and all these million base pairs are sequenced parallelly at the same time. That's why it's also called as massively parallel sequencing, right? And then the beauty is the computer, it matches. Okay, the first bit goes and aligns to this region, second bit goes, so millionth bit goes here. Now I have the data, I have all the genome of the patient from chromosome 1 to chromosome 22, X and Y in case of a male. So I have everything. So the entire genome library is like this, and what I do, the bits and pieces, yeah, this is aligning here, is aligning here, fine, awesome. So based on this alignment I get everything. Okay, so the entire alignment is done. This is how the sequencing is done. Okay, for example, this picture, see, this is a reference genome and these are the things which I said. Like here it is, these are the I drew a line like this and I initially cut, right, into different segments. These are the segments, first segment, second segment, third segment, those segments which are aligning to this segment has a match here, this segment has a match here, this has a match here, so it has aligned to its position, right? Thus I will get the entire genome sequencing. So what is the use of it? Why should I break my head doing this? Yeah, I know Marfan syndrome is a mutation in fibrillin gene, I go with that. I know P53 is mutated in this type, I go with that. Why should I break my head? Why should I break my head doing introducing a costly technique and all these things? Is the use. First is targeted sequencing. When you read cystic fibrosis in a chapter in Robbins, it will be mentioned the most common mutation, that will be the question asked, but there will be also said there are more than 200 mutations identified, right? So if I look only for this may be accounting to 75 to some percentage. If I'm looking only for this, I'll be missing out on all these things. That's where my next generation sequencing helps. Cystic fibrosis entire CFTR gene can be amplified in next generation sequencing. This method is called targeted. I have a target. I want only CFTR gene. I want only genes involved in glycogen storage disease. Target, I have a target, so I sequence only them. That's called a targeted sequencing. Next is a whole exome sequencing. This comes to a beauty. So here I can identify both known as well as unknown disease. For example, we'll first come to the known disease. Okay, we all know retinitis pigmentosa is a genetic disease, but if you look at the genes of retinitis pigmentosa, it is like n number of genes are involved. So for that, it will be easy and cheaper for me to go with whole exome sequencing. Exome accounts for about 1.5% of the whole genome, of which only I sequence those parts. How will I sequence those parts? Because I will design the primers, which I said before, I will design the primers only for them. So only the exons will be amplified. If only those exons will be amplified, they will align only to the exonic region. So that's called an whole exome sequencing. Unknown diseases. You are a pediatrician. Imagine yourself, you are sitting in a clinic. A patient comes with dysmorphism, lots of features, aplastic anemia, blah blah blah. You can't fit into any disease condition. You don't know it's any disease condition, or you presume that it's something new, something novel. Then you go for a whole exome sequencing. I'll be able to give you an answer. Yes, boss, this is the mutation. You may go ahead and name it after your name because it's the first case, or you can just use it for treatment. Then where does whole genome sequencing comes is, as of now, whole genome sequencing is used only for research purposes. The use of whole genome sequencing will be epigenetics. Very, very important. Especially in cancers, this is coming up in a big way. Epigenetics. So this is exome. What is epigenetics? I have talked about epigenetics in my previous videos. If you have not listened to them, this is a gene, an exome. So before this gene also, there will be some DNA material. This guy will tell, yeah, you go ahead and produce a protein. Or this guy will tell, no, you stop. This is not the time for you to produce the protein. This guy is called epigenetics. This guy controls the gene. That is epigenetics. So I will be able to say this guy is at fault only when I do a whole genome sequencing. Okay. But as of now, whole genome sequencing is used mostly for research purposes only. So what are the disadvantage of or which of these test NGS which diseases can't be detected? First one, as I said in the previous video, it's a DMD. So what is a DMD? It's Duchenne's muscular dystrophy, where the deletion is 1.5 megabase in the dystrophin gene, right? So fine, we'll look again the concept. It's very easy. What I do is I cut in bits and pieces, right? So if what I told is approximately 250 base pair. If 1.5 base pair is deleted, I won't be able to say, I won't be able to cover. Maybe one half will be there, or this some half will get aligned to that place. No, so I won't be able to tell that it's a deletion. Okay. Second one, CNVs, which is copy number variation, which means for a particular person this sequence is repeated 20 times. For someone else, the same sequence will be repeated 200 times. Will I be able to say? Not at all, because I am using the method of amplification. So if this segment is there 1 times or 200 times, it's going to get amplified. This will be amplified. So copy number variations, as of now we are not able to detect, but there are technologies coming up also. Third one, translocation. Can I detect a translocation? No, not at all. I am breaking up into 150 to 200 base pair. If it is 922, anyway it's going to get translocated from 9th chromosome to 22nd chromosome and vice versa. So when I break it, I won't be able to know whether this is attached or not. But when I break it, both 9 and 22 will be there. Yes, they will be there. So this will go and get aligned to chromosome 9. This will go and get aligned to chromosome 22 irrespective of the translocation. So I can't identify translocations. The main disadvantage is a large deletion or duplication cannot be identified. Copy number variations cannot be identified, and translocations cannot be identified. This may be the question which can come in the future exams. So just one passing mention about GWAS. What is GWAS? It is Genome Wide Association Studies. It's based on SNP. If you don't know what is SNP, it is single nucleotide polymorphisms. So what is single nucleotide polymorphism is? There are millions of people. Each individual is different, right? So what makes each individual different are variations in the genome. Of this variation, one most important thing is SNP. SNP is one nucleotide is altered from the reference. For example, I am there and you are there. You means the person who's watching the video, right? So I have in chromosome 1 nucleotide is T for me, but similarly for you in that area it is a G. This is actually a mutation, but this thing will not affect anything. You are also normal, I am also normal, and this change will be present in greater than 1% of the population. Okay, this is called single nucleotide polymorphism. Fine. GWAS is based on single nucleotide polymorphism. First thing you have to identify is all GWAS are association studies, cannot be used for diagnosis until and unless proven. Okay, what is GWAS is, okay, we have you will sit at a diabetic clinic and you see 1000 cases per day. Okay, we'll see over a month. Per day is too much, right? Yeah. So over a month you see 1000 cases. You collect all the 1000 persons DNA. So you will get 1000 people's data. So you see these polymorphisms in all the 1000 people. Okay, and you get 1000 normal individual. Same way you do everything. So you see what are the polymorphisms, what are the SNPs which are present in these 1000, which are not present in these 1000. Then you can say to some extent with statistics, this is associated with diabetes mellitus. That's how all these all the HLA are association studies. They are just associated with diabetes. It need not cause diabetes. It means if by statistics you have this gene, the probability of you getting diabetes is more. That's all. That's all you know about GWAS. One thing you have to know, GWAS is always an association studies. You carry out throughout your clinical practice. You can never say that person is going to get the disease based on GWAS study. It's very, very important. And most of the polygenic disorders where more than one gene is involved, or complex disorders like diabetes, hypertension, obesity, All these, for all these conditions, GWAS is being carried on, and which may give us clue in future to pinpoint a gene as a causation. Thank you. If you have any queries, you can ping me on my page or on my group, on help group. Thanks a lot. Have a nice day.